tissues is epithelial tissue. Epithelial tissues are interesting because they're a connective sheet. They wrap around things. Can you think of some organ in your body that would be like a sheet that wraps around things? It's the largest, over space, it's the largest organ in the body. The heaviest organ is actually the liver. So if you're saying large by weight, it's the liver. But your skin, yeah, your skin can actually be really light, but it covers so much surface area, we consider it the largest organ in the body. So your skin is made of epithelial tissue, but that's not the only thing that epithelial tissue is. It's any kind of wrapper, and every organ in your body actually has a wrapper. Your brain has a wrapper around it. We call it the meninges. So if you get an inflammation of the meninges, we call it an itis, so it's meningitis. Your heart has a pericardial sac, or we call it the pericardium. It goes around your heart. If it gets inflamed, guess what they call it? Pericarditis, right? Your lungs, we call the pleural sac. So if those get inflamed, you have pleuritis. So your whole abdomen, all of your internal organs, like your stomach, your small intestine, large intestine, have this blanket-like sheet that goes around. It's called the peritoneum. If it gets inflamed, let's say your appendix bursts, you get peritonitis, because that can get inflamed and really nasty. But epithelium are always sheets. They're like kind of like Legos that connect sideways, and they can connect top to bottom. So they form some kind of a covering. The really tricky thing to think about is, where does your skin end? It doesn't. Your skin's continuous. It goes all the way up your body, up and in, into your mouth, and actually folds over, and it lines the inside of your mouth, right? It wraps down around the teeth, back up, goes over around your tongue, goes down your whole throat, forms your esophagus, down to your stomach, to your small intestine, your large intestine, and then out, and then back up around your body again. It's one continuous sheet, no breaks anywhere in that sheet. It's always got to be continuous, because if you have a break in this epithelial tissue on your skin, what would be a problem? You can get bacteria, and it could be an infection. You could lose water and fluids, which could make you dehydrated. So there are a lot of things that could happen if you break this sheet. We're basically a water bag that's continuous and has a tube that goes through the water bag to let food pass through. So epithelium is also an easy one, because the way we name epithelial tissues are by what they look like. So if we're naming, here are some examples: blood vessels, epithelium; the lining, tubes in the body; the lungs, airway, lining; your kidneys, your small intestine, your large intestine, all of those. You can see they have special shapes to them depending on the function. So how we name them, oops, let me click on this, right? So if we have the number of layers, if it's only one layer, we call it simple. If it's two or more, we call it compound or stratified, so typically stratified. And then the second way we name it is the shape of the cell. So the first name, it's like you get two names. You get a first name and a last name. So do epithelial tissues. Their first name is the number of layers, the second name is the shape of the cell. So if I have these little square or flat, you can see how they're squashed, right? We call them squamous, and I always think squashed squamous. If they look like cubes or boxes, we call them cuboidal. If they look like tall columns, we call them columnar. Sorry, my focus was getting off. So your shapes are either squashed, boxy, or columnar, right? So if I go through the same list again and I look at lots of these sheets, so at least two or more layers, 1, 2, 3. I'm looking at the round circular thing in the middle. That would be the nucleus, right? So I have 3 layers here. That's definitely stratified. And I look at the top layer. This is where it gets tricky, even for med students. The bottom layer may look more square, but it's the top layer that counts. So the top layer is squashed, and we call it stratified for many layers, squamous. Okay. Next, cuboidal. So I have 1, 2, stratified, 2 or more layers, and they're like boxes. My top layer looks like boxes. It's nice that the bottom layer does too, but it's the top layer we depend on. Because you can see lots and lots of stacks of cells that might be different shapes, but it's always the top layer that we look at. The next one, stratified columnar. We have 1, 2 going up. So you have 2 layers, it's got to be stratified. Columnar looks like 2 columns stuck on each other. Right? And then we can have different types of combinations. Like we have some that say pseudo-stratified, because pseudo means false. It's not actually stratified, like in this example over here. Pseudo means fake, stratified means it looks like many layers. Because it looks like you've got a layer of nuclei here and a layer of nuclei here, but the thing is, look at the cell. This cell actually reaches all the way up to the top and all the way down to the bottom layer. This cell that looks like the nucleus is here is the same way. It touches the top, but its cell body goes down to the bottom too. The nuclei just shift around so that they can get closer together, but it's actually only one layer. It just looks like 2, so we call it pseudostratified. All right, let's look at some slides. So we're going to do this. We're going to look at your skin. Well, not your skin, sorry. We're going to look at some skin as an example. Let's go over to this. You're going to pull out number 14. Right? So make sure you pull out your other slide. Right, human blood. Went up here in slot number 21. Now we're going to pull out number 14. Ooh, I missed it. It looks like 14's here. There we go. Squamous epithelium, WM. Do you remember what the WM stands for? Whole mount. Okay, so they took a piece of tissue, boom, they stuck that chunk down there. All right, I'm going to turn my revolving nose piece to which one? The red. If it feels loose, just give it a little twist to tighten it back up. Mine just felt a little loose. There's my red, which is my scanning objective. It's 4 times magnification. Right. I'm going to put this back down so I can have 2 hands. Push down on the spring clips. Bring it in. All right. I'm going to pull that right so that little red spot is in the center. It's been stained. I'm going to make sure that my camera is on. Ooh, that looks pretty dirty. I'm going to pull that right back out. And my tissue. Just going to give it a wipe one direction. That looks pretty clean. And I'm actually going to take this and wipe off my stage while I'm at it. There we are. Just push it right off the edge. Just like crumbs on the kitchen table. My wife, she'll kill me if I said that. Even though I do pick up after myself. All right, there we go. It's not perfect. So what are we going to do? We're on scanning, so you can use the coarse adjustment, but this is really close, so I'm just going to use the fine adjustment. Oop. Wrong way. Looks like my surface is a little dirty still. Smudged at least. There we go. Very cool. All right. Okay. So right now it looks just like a sheet. Came into pretty decent focus. I'm going to go to my revolving nose piece. I'm going to turn it. Does anything look clearer to me? No, it does not. There we go. Okay, here's where it gets tricky. So what happened, why am I not seeing? If it's simple, squamous, it should look like squashed cells. But these all look really uniform, right? They don't look squashed. I mean, they look like they're spread out. I'm going to get a little bit closer because right now, I don't know if you can see it really well, but I have all the dots, which are what? Those are the nuclei, and then around the dots, I can see a very light pattern. So I can kind of see like a light pattern going around. Boop. Each of them. fine tuning here. There it is. Oh, I went right by it. Slow, slow. Oh, I just missed it. There we go. It looks like I'm right at a fold or something, so I'm just gonna pull up. Oh, another fold. It's not very dark. I like the darker ones. Down in here. Wrong way. Down in here. Oh, that looks great. Autofocus on my camera. I keep forgetting to turn that off. I'm gonna see if that really helps. Let's see if I can use autofocus to my advantage. Just pick some. Oh, that's not too bad. Let's pick this guy. See if I can pick the crack right in between. It's okay. I'm turning the filter on the bottom. I'm going to give it another turn. It's not too bad. It's pretty interesting. Right. I'm turning the rheostat too. I'm darkening it up a little bit. Alright, I'm back to the first one again. So no color added. I don't like that. I'm going to go back. And you know what? You see those cells on the top? Let's see what they have to offer. Oops, coming down. Oh, that's the edge. That's why they're pretty clear. A little more right here. That was decent. Okay, where I'm going to focus on is down here, kind of right in this area. Do you see them? So like for instance, this cell right here, right in the middle. See its shape? It's kind of like a pentagon. So if I were to draw that, these cells go around. I'm not going to draw them all, but I'm noticing kind of a pattern. And this is kind of like the human cheek cell. Remember when we scraped your cheek? I've got a little square, square. So strange not looking with one eye and drawing with the other. And then right in the middle is the nucleus. And then right next to it, I have another one that has kind of an odd shape because it looks more like a tall top. Also had it. Yep. And this is kind of the same way. Has a nucleus. Just draw a couple of them. I'm kind of noticing it's not a perfect pattern, but it's kind of a pattern. So here's where it's tricky. If I look over here at my manual and I see simple squamous, because that's what we're looking at, right? Simple squamous. It should be one layer, but it looks like there's thousands of cells there. The thing is, look at the perspective. If I look at it from the side, I would see one layer all the way along. There's another one, and there's another one, and there's another one, like this. What's happening is that I'm looking down on it. So each of these cells, I'm seeing the longer or the wider perspective. So that's what I'm seeing right here. I'm seeing each of these from the top perspective. Oh no, for some reason my drawings didn't come out. I'm sorry. Let's see if I can fix that quickly. Go without spending too much time on it. Is that better? Okay, perfect. Sorry, there were my drawings right like this. And if I looked and I drew the next one, it just disappeared, but I remember, and I think you could probably figure out the pattern. They're like that. So if I imagine the cell from the top, I might see the squash cell with the squash nucleus here, where it looks kind of like a long nucleus. And then I see another one here. Where the nucleus is at is important, because in simple squamous, or squamous in general, you're going to see the nucleus smashed, but it stays in the center. Because this can be confusing. Sometimes you'll look at a columnar cell, and the columnar won't have the nucleus in the center. It'll have the nucleus kind of squashed looking, but sitting towards the bottom, or what we call the basement membrane. Basement membrane is what holds this tissue to your body. It's the glue that holds it down. So if I were looking at this in 3 dimensions, I would probably see something like that, right? So if we're looking from the bottom down, I'd see more of a round image. Same thing with this one. It's like going to the aquarium. Here I'm standing right here looking, and I can see the animal here, but if I were standing up above, I'd see the same animal, but I'd just see a top perspective coming down. So then I'd have another cell here, that same thing. And that's what I'm seeing. It's from the top view, but if I look from the side, it's a sheet that's laying on that basement membrane. And that's how epithelial tissue works. If I'm looking at skin, different layers of skin, I'll see cells that are sometimes darker than the others and sometimes some that are more clear. Because here's the basement membrane, stratum basale, the basement layer, Latin. And then you can see as I'm coming up, they change a little bit. And we'll see a picture of this actually later on. But I can see other cells like a Merkel cell or a Langerhans. This is your immune cells. This is for sensations. A melanocyte makes a chemical called melanin that gives our skin color. So melanin is kind of like paint. We have a spectrum of melanin. You have more of one color, you have darker skin. You have more of another color, you have lighter skin. You have none, you have no coloring at all. You're an albino. But that's what this does. Melanin is made down here at the basement, and it's like, well, it's like food coloring or whatever. It leaks up towards the top. The closer you get to the surface, the less of anything. Notice these nuclei, they disappear. Because, and this is really important, epithelial tissue, the more layers you have, the harder it is to get nutrients through. Your skin is very thick compared to other cell tissues. So we make our coloring of our skin here, and towards the surface we have no coloring. It's like clear little shingles that you can see through and see your true skin color. But if I were to peel this off, there would be no color to it. It would be like well, you've had a sunburn before probably, and you peel that off, and that layer is clear, right? It's like when you put glue on your hands and you let it dry and you peel it off. Look, my skin came off. Same thing. So it doesn't matter what your skin color is, that sheet will be clear because that's the layer of dead cells that are not getting nutrients. And that's a whole other topic to talk about when we get into skin coverings. All right? Let's move on to the next tissue. Remember, there are 4 tissues. Number 1 is epithelial, and every time you see it, you have skin coverings. I'm sorry, you shouldn't say skin. Epithelial is always covering something. Epithelial tissue or epithelium, always. Connected side to side and always covering.